Welcome everybody to this event in the context of the GVN Forefront in Virology webinars. And I will speak in a minute about our guest speaker, Matthew Frieman, but just let me explain, as always, for those who maybe are not familiar with what the GVN is. The Global Virus Network is a network of about now 65 research centers all over the world and 11 centers. This is really a network which is science-based, which is independent, and these are obviously important keywords, merging really the best virologists worldwide, working on all viruses, on all human viruses, but obviously very much focused nowadays on COVID-19, but really working on all human viruses. And our missions combine research through task force, through annual meetings, education, a lot of program, and in particular in the context of the GVN Academy, which has now been initiated, and we have activities of mentoring, of fellowships, support of Rising Star, and finally advocacy. At the end, it's also about partnership with the industry, partnership between academics and with a number of national and international institutions. And webinars are an important component of our activities. And in this context, it is really my pleasure to welcome Dr. Matthew Frieman. We are very grateful, Dr. Frieman, that you took on your time for this lecture. Dr. Frieman has been educated in St. Louis, in the Washington University of St. Louis, and then in Baltimore, in the Johns Hopkins University School of Medicine. He moved from 2004 to 2009 to get a postdoctoral fellowship at the University of North Carolina with Dr. Ralph Baric, that we know well at the GVN and who also gave very important webinars. And then from 2009, he has been working at the University of Maryland School of Medicine as an assistant professor, and then an associate professor, and now a senior. I should mention that he is also an affiliate member to the Center for Vaccine Development and Global Health, and an affiliate member of the Institute of Human Virology, headed by Bob Gallo, who is the founder of the GVN, and as I said, part of the University of Maryland. His work has been very much focused on coronaviruses. I'm not going to get into detail, he's going to present, but he has produced very significant results, and we really look forward to your lecture, Dr. Frieman. Thank you again.
Thank you very much for having me. I'm excited to present some of the work we've been working on in the lab. I'm certainly open to questions for anyone who has questions as we go along. I will try to answer them if I see them come up, or we can answer at the end. And thank you again for having me. I am here at University of Maryland School of Medicine. And the goal, just as a bit of a background to what the lab does and on coronaviruses, is that we really have started here since my postdoc, understanding how SARS-1, and then when following that MERS and now SARS coronavirus 2 really cause disease, and then what can we learn about how they cause disease on to understand how to inhibit that disease. And so we've done this for now 12 years now since I've been at University of Maryland, trying to understand at that point SARS-1 and MERS, but now layering on all of those models and knowledge that we had previously onto studying SARS coronavirus 2. Let me get my pointer set up here. And so we use a whole set of models in the lab, from looking at in animal models and reverse genetic systems, all kinds of cell culture models, from standard cell culture models to long organoids and long chip systems, virus-based or yeast-based screening using genetics, which a project I won't talk about today, and also in virus transmission work with Don Milton at University of Maryland College Park. And we do all of our coronavirus work in these systems, as well as dabble in influenza virus as well. And so I'll just share with you a couple of the stories that we've been working on in the last, I guess, almost 2 years now for SARS coronavirus 2 today. And so everyone is a coronavirus expert. I don't think anyone needs to know this at all, but I put in a couple intro slides in case you have been living on an island for the past 2 years and didn't have internet. There are 7 human coronaviruses that we know of now, including SARS coronavirus 2. The 3 that are BSL-3 pathogens are SARS-1, MERS, and SARS-2. They all have the same structure, which is basically envelope. They're envelope viruses with these spike proteins on the outside of the virus here in the cartoon. You can see them in the EM picture on the right-hand side here. These spikes are the immunodominant antigen, and it's where all of the vaccines are directed. We'll talk a little bit about vaccine work at the end of this talk as well. If you look on this phylogeny, this is an older phylogeny. SARS-2 wasn't on here, but I like it because it shows, one, that you can see the other 4 human coronaviruses give you a common cold are in these alpha and beta coronavirus lineages, with MERS up here on the upper left-hand side and SARS-1 over here on the right-hand side. And interspersed between all of these lineages are these bat coronaviruses, which we now know are progenitors for almost all known coronaviruses that we can tell of at the moment. And when SARS-2 emerged, it was clearly a SARS-1-like virus. It has shown out to be around 1% lethal in humans. SARS-1 is 10% lethal, infecting 8,000 people in 2003 when it emerged. And MERS is still quite lethal, 35% lethal, and there's still MERS cases all the time, mostly in Saudi Arabia. But now that we're in the middle of our SARS coronavirus 2 pandemic, we really are in this 4th stage, which is driven by the Delta variant. We'll talk about variants in a slide, but really what I want to point out from this slide is that we still are certainly in the midst of this pandemic. Just because we have vaccines and we're trying to get about our business doesn't mean this virus is gone. And so when we think about where we are now in research and therapeutic development, we have a ways to go. As the virus has emerged since the late 2019, there's been a whole range of variants that have also emerged over time. This is images from NextStrain. I think it's easiest to show on the right-hand side here. You can see that all the way from November 2020 all the way through now, we see this really huge wave of variants that have emerged. Initially, it was Delta over the winter and spring of 2021, and that gave way to this so-called Delta lineage, which is really a composite of a lot of different viruses, all in this called Delta by the WHO, but that has really taken over much of the world's infection by at least the ones that are sequenced, including some of the new variants off of Delta, which are now starting to emerge as kind of riffs off of the Delta theme. This is data in the US from the CDC showing the Delta lineage, which the major isolate from Delta is called B1351 which is the Pango lineage. In the US right now, it's around 99.5% of all isolates that are identified in the United States are this B1351. And I think it's certainly important to understand what it is about this new variant that is causing this dramatic spread. And we're not going to talk about that today. I'm happy to talk about that in the questions at the end if you'd like. But certainly just to show that we certainly are in this wave of this new lineage that I think is certainly important both for vaccine and therapeutics as well as for limiting transmission, which it seems to transmit incredibly well compared to the previous isolates that we had in 2020. So back to kind of the origin story here of where these viruses came from. So all the coronavirus emergences were first identified really on ProMed. This is the first place that anyone heard of SARS coronavirus 2, or at that time just this novel coronavirus. And so SARS coronavirus 1 emerged in 2003 in the Guangdong province. MERS emerged in 2012 in Saudi Arabia initially identified by a very observant clinician, Dr. Zaki. And then SARS coronavirus 2, as we know now, the first announcement was in late December 30th on ProMed from this cluster of pneumonia cases that were identified in the Hubei province in Wuhan. And if we think about all of the emergence of the coronaviruses we know of, they all are really linked to zoonotic cases. And so there are certainly a lot of drivers of zoonosis, whether it's wet markets or encroachment into the environment in forest where humans normally hadn't been. Clearly, food scarcity and climate change is driving animals and people together more than they had previously been in centuries before, and also just time. So the more time we dwell in the environment with animals that can harbor these viruses, really the bigger more bigger propensity we are to be exposed to them. And so this leads all to the kind of therapeutic drive that we're doing in the lab, where we really have to prepare now for these future zoonotic events. And so this includes developing pan-coronavirus antivirals, because we really want to identify therapeutics that work not against just the viruses we know of now, but the viruses that could come in the future. And so we have a large cohort of large set of projects looking at pan-coronavirus antivirals, at broadly acting antivirals that are targeting the host, looking at vaccines and antibodies that can really target all across known coronaviruses, and really also taking it back to the cell biology and pathogenesis step, and really understanding the pathogenesis of these viruses, both in animal models and in humans. If we look back at the replication cycle of the virus, what we find is that it replicates in kind of unique ways compared to other RNA viruses. So you have attachment at the membrane, which is normal for any virus. You can have entry right at the membrane based because of protease cleavage of spike, or endocytosis into the endosome. There's unique aspects of the transcription cycle leading to these subgenomic RNAs, as well as a whole host of viral factors that regulate the cell biology of the cell. So affecting everything from innate immune response to MHC presentation to lipid modifications, and all of these are required for the virus to replicate efficiently and cause disease. And so all of these aspects of the virus life cycle are certainly druggable. This is from a review with Julie Dial that we wrote several years ago after MERS emerged. And all of these are true for SARS coronavirus too, and really all the coronaviruses. So we're really trying to understand the basic cell biology of these viruses better, so we can really develop therapeutics that will get us there in the future. We've done a lot of that therapeutic development now in the lab over the last 2 years. I divided up here basically kind of on the places in the virus that in the life cycle of the virus that are therapeutically targetable. And so we've done a lot of work on antibodies and vaccines from monoclonals with Regeneron and AstraZeneca. Novavax, we've been focusing on their vaccine, as well as with Overlevy and the SAVES group on looking at adjuvants and other immune variants that are viral variants that are important for emergence and spread of the virus. We've also been working with, so I'll tell you about the Novavax project second today. We've also been working with Bill and Melinda Gates Foundation on antiviral development, working with Sarah Cherry through that program. When I'll tell you about a antiviral project we have with them, but also working with Pfizer on their new protease inhibitor that targets the one of the viral proteases that's expressed during replication, as well as looking with BARDA and DARPA with Don Ingber and NIH looking at additional antivirals there. And then before SARS-2, we were very interested in host factors that are important for replication of coronaviruses and other viruses too. And we've been well funded by this group called Keto Pharma, which is funding a development project now looking at the SKI complex, which is a RNA degradation complex in the cell. We've identified drugs to that complex that inhibit virus replication across coronaviruses, filoviruses, and influenza, as well as additional project with BARDA and DARPA there looking at host factors. And if we look at all the therapeutics in general, it really doesn't matter what we develop, the timing of their administration is really important. And so we're trying to understand more about the host disease, the human disease in the place of SARS coronavirus 2, which we really couldn't do for SARS-1 and for MERS, just because of the number of patients that were available. And certainly the development of antivirals is really important for administration very early in infection. If you give it late, it doesn't work. That's been shown over and over again in trials for even remdesivir and molnupiravir, which are currently approved or almost approved antivirals that we'll talk about next. So they all have to be developed, have to be given early for there to be really a chance of them to work. Once you get into dialysis and you need more anticoagulants, you really are, it's a problematic time in this, and antivirals, generally the virus is gone or the damage is done already. And so we really want to develop antivirals and other therapeutics which can really be used across these boundaries by looking at both antiviral effects as well as anti-inflammatory effects that are able to be used in vivo. And so I'll tell you about the first project with Sarah Cherry at Penn. This was funded through Melinda Gates Foundation, and the project was run as our PM by Mona Lisa Chatterjee. And so what Sarah did at Penn was early on in the epidemic, they started screening tens of thousands of drugs through various libraries, looking for drugs that were able to inhibit SARS coronavirus 2. And they developed a bunch of cell models. They ended up using Cali3 cells, which Sarah has found really have a really strong response to antivirals and much more in vivo-like phenotypes in her assays. And she's identified a whole host of over 100 different drugs that are able to inhibit SARS coronavirus 2 in one way or another, and they can be grouped into different pathways. One of the largest pathways that they set of drugs they found were nucleoside analogs, and certainly for her through screens, she found just by through the blind assays she was doing, she identified remdesivir and at that time called EIDD-2001, now called molnupiravir for Merck, as part of her screen, but a whole host of other nucleoside analogs that were very effective in her Cali3 system. When she started looking at the other, at some of these analogs in a variety of cells, one, she found that some of them worked better in some cells than others. So you can see thioguanine here works very well in CALUs and A549s, but not as well in KICOs and H877s. And you can kind of see some differences here. And actually there's interesting biology, we think, that certainly can be followed up by Sarah, and we're going to be trying to understand it in vivo about which drugs work against which pathways and which cells, trying to understand about mechanism of action of these nucleoside analogs. They're not all the same. Many of them are purine analogs, some of them are pyrimidine analogs, and based on the pathways that the cell uses to produce purine and pyrimidines, that really drives what therapeutics we should be developing. One of the ones that she found was something called Brequinar, which is a DHODH inhibitor. It's involved in pyrimidine biosynthesis, leading to the pyrimidine nucleotide, which is used for replication of the virus and the cell in general. And when she started combining, what she did in Cali3 cells was start combining Brequinar and EID-2001 or Brequinar and remdesivir, looking for synergy. She did a whole host of these drugs. I'm just showing you the figure from the paper, which is Brequinar against these, as well as this BAY, this Bay drug. And what she found was that there actually was synergy. And most drugs, you don't see synergy across these. So this is another drug on the bottom that came out of the screen, AVN944. It doesn't show synergy in this Bliss analysis, which is what this highlighted graphs are here. So blue in this graph is showing, in these tiled diagrams are showing synergy. She was also able to look in air-liquid interface cultures from human bronchi and primary epithelial cells, and she was able to show that EIDD alone, either the Bay drug or Brequinar, the Bay drug alone didn't have much antiviral effects. Brequinar did by itself in this system. EIDD-2001 certainly did as well, shown here in the third bar here. But now if you start dosing down and you add in Bay and Brequinar, you can see there's actually a synergistic effect, even in these air-liquid interface cultures, which are much more human-like than the Cali3 cells. So this led to our studies where we're really looking at what happens in vivo and trying to study these antivirals in a mouse model. And so I won't go through all the mouse model development we've done in the lab, but certainly there are all these factors that affect replication of SARS coronavirus 2 in humans and in mice. The variables we've identified so far that certainly affect replication and pathogenesis of SARS-2 are age, comorbidity, and immune status, also genetic background. All of these affect the severity and type of disease that we see in the mice. We've also found, and as many people have found, that the ability for these viruses to replicate in mice is certainly important. And the original isolate of SARS coronavirus 2, the Wuhan 1 or the Washington 1 isolate that we use in the US as our control virus, doesn't replicate in mice because it doesn't bind to mouse ACE2. Viruses needed to adapt to the mouse ACE2, they developed through bypassage or by variants in humans, a mutation at the spike protein at 501Y, which allows for spike to engage the mouse ACE2 protein and allow for replication. And just so by chance, many of the, and some level of selection, many of the isolates that are, the variants that are emerging from the original UK isolate, the B117, through South Africa, Brazil, the Cal3, New York, Peru isolates, all of these variants actually have the 501Y mutation in them, which allow for them to replicate in wild-type mice. The Barrick lab and several other labs developed mouse-adapted viruses, which replicate in wild-type mice as well. We use the MA10 virus, which I'll mention at the end. And so we can use wild-type mice now instead of having to use the K18 ACE2s, which overexpress ACE2 in almost all cells of the mouse, leading to lung infection, but also encephalitis in the brain, which isn't really the levels that we're seeing in humans. And across all of these viruses, we've done validated the strains by sequencing them, done inoculation curves looking at dose-dependent effects on pathogenesis, and looking at various time points where we look at lung titer, RNA, lung histology, and inflammatory responses. And all this work is ongoing as new variants emerge. And the reason I'm focusing on this is because it's important to understand the dynamics of the infection and the model that we're doing, so we can really develop therapeutics and test therapeutics in the lab that are really the more applicable to humans use. And so just showing you as an example, two of these models here. This is the B117 and 1351 infection of BALB/C mice, typical BALB/C mice. On the left-hand side here is weight loss. So neither of them have much weight in the 10-week-old mice. But when we look at the lungs, what we find is that even though both of these viruses have N501 mutations, B117 replicates quite poorly, a little bit in the lungs at day 2, it's gone by day 4. But B1351 replicates incredibly well in the lungs, upwards of 10 to the 9th PFU per gram lung by day 2. They both have incredible inflammation in the lungs, with B117 actually having a bit more, even though it doesn't replicate as well. And so we're trying to understand these differences as a way to understand the dynamics of pathogenesis as they go through both natural host studies, but as well as for our therapeutic development. So the model we settled on to initially study a lot of these drugs through our Gates Foundation work and Sarah was the B1351 virus. And so this virus does have the 501 mutation. It has several other spike mutations in them as well, and a couple outside of spike mutations, which we think are probably important for disease going forward. And so in the initial study I'll show you, we infected 10-week-old BALB/C mice with 10 to the 5th PFU of this B1351 isolate, which I believe we got from Andy Pekosh's lab at Hopkins, sequence verified, and then used in the animal studies. All of the dosage curves I'll show you started at day -1, so they're prophylactic doses. We start all of these early before infection. We infect at day 0, and then we harvest mice at day 2, looking at lung titer and histology using the models that I just showed you. And the first study, what we did is we performed a dose curve of EID 20:1 with starting at 150, 50, and 16 mg per kg. All this is oral dosing. And then we combined that dosing with either Brequinar or the BA molecule to see if any of them inhibited or synergized with SARS, with EID 20:1 in vivo. And so if we just look at the lung titer in these mice, you can see that on the left-hand side here, EID 20:1 certainly has a dose-dependent decrease in lung titer. If this is just day 2 time point here. When you add in Brequinar, you can see a little bit of an effect at the lower dose, so about a half a log to a log dose decrease in the virus in the lungs. Brequinar by itself had no effect. The BAY drug, the BAY drug, had no change in titer at all, even though it was quite effective in vivo. In vitro and the ALI cultures. The really interesting part of this for us was looking in the lung pathology. And so I'll just walk you through what we see here. So on the right-hand side on the top is no infection. So this is a nice open airways, nice alveolar space that's open, and really limited damage or no damage at all in the large airway in the epithelial where the ACE2 is expressed. Well, if we had no drug on the left-hand side here on the upper left, this is just virus alone. It's certainly quite inflamed. You can see there's a mixed inflammatory cuffing around the bronchi and the and the blood vessels and the arterioles here. We see edema around the lungs and denuding bronchiolitis. So we can see sloughing of the cells in these large airways. As we add in EIDD alone on the left-hand side here, there really isn't a difference. Only when you get to the very high levels of EIDD do you start seeing a little bit of a reduction in inflammation. So while EIDD 20:1 is certainly a potent antiviral, it doesn't have much effect on the inflammatory response, at least in our experiments. What we find really quite interesting is that when you add in Brequinar, even at this low dose of EIDD, and low dose of Brequinar, 20 mg per kg, we see a really dramatic effect on inflammation. And as you go a little bit higher into 50, at the higher doses of EIDD at 50, 150, you really see this dramatic anti-inflammatory response that is dose dependent and seems to correspond with the antiviral effects that we're seeing from EIDD as well. And so what we want to do is push this a little further. We went to a higher dose of Brequinar across a larger dose curve, but we kept EIDD constant in this experiment at 50 mg per kg. It's kind of moderate dose. So we didn't want the molnupiravir EIDD 20:1 to be incredibly potent. We wanted it to be in the middle range so that we could look for enhancement and reduction and synergy in vivo. And so we set the experiment the same way. We had a, oh, that's the wrong schematic there. So we had the EIDD at the same concentration, but increased the amount of Brequinar. And so Brequinar alone on the right-hand side had no effect. It looked like virus alone or carrier alone in this experiment, carrier plus virus. What we find is that EIDD alone has about a 2.5 log reduction in virus titer at day 2. But now when you layer in EIDD 20 or Brequinar in this dose curve, you can now see an enhancement of antiviral effect compared to the EIDD 20:1 alone. And importantly, we also saw an effect on inflammation. And so again, like before, the EIDD 20:1 on the left-hand side here shows significant inflammation in the lungs. When we have no virus or no drug alone, we see a little bit more inflammation. But Brequinar certainly still had anti-inflammatory effects. We actually see a strange effect at the high dose. I think we're getting to toxic levels here on Brequinar, but at the lower doses, we again recapitulated the phenotype we saw before. And so this certainly shows that we are seeing this dual effect from this combination therapy, which we think will be useful potentially in the future. Now that molnupiravir has finished its phase 3 trials, we're working with Gates Foundation to forward this combination therapy into future trials as well. And so from this initial part, I wanted, hopefully you got that there were, we've been able to use in vitro screens, both my lab and Sarah Cherry's lab, to identify a whole range of compounds that we are now looking for synergy in vitro and synergy in vivo. That the combination of EIDD and Brequinar is able to inhibit replication in the lungs and reduce lung pathology. Again, 3051. I didn't show you the mouse adapted virus data, MA10, but we have the same effect in the MA10 model as well. And what we really believe is that now there certainly needs to be more antiviral development to really combine drugs together, and maybe similar to what is done for HIV, you need a combination therapies to hit the virus in multiple pathways to drive antiviral effects and have a strong therapeutic effect in vivo. Okay, so that was the antiviral side. I want to show you a little bit of the drug work we're doing, the vaccine work we're doing as well. And so we've been working with Novavax on their vaccine since early 2020. We've been working with them since 2015, actually on their MERS coronavirus vaccine. And they use the same technology for MERS that they've now done for SARS-2. And this is an old MERS slide I just wanted to put in. Basically, the schematic that they use for, the scheme they use for their vaccine is shown here, where they take the spike protein off of the virus, they express it in by baculovirus in insect cells, and then they can extract the full-length spike off of the surface of insect cells and make what they call nanoparticles. And so when they mix it with adjuvant. And so what you get is a little floret of spikes together that produce a very strong antigenic response in vivo. All this is combined with a matrix M1 adjuvant that they have worked on in the company and they own. And this is from this Chilean soap bark tree It's a saponin-based adjuvant. And the importance of the adjuvant is been able to be seen in all of our experiments where protein alone doesn't have an effect, a very strong immunogenicity effect. But when you start adding in the adjuvant, now you get a really heightened antibody response. And just showing old data from MERS, we did this for MERS before. If you vaccinate mice with the MERS vaccine, and then you challenge the mice with MERS in these old experiments, you can certainly see a reduction in virus load in the lungs. And so we did the same thing with them for SARS coronavirus 2. One of the interesting developments in the field over the time period between MERS and now SARS-2 was really the pioneering work of Jason McClellan and Barney Graham, where they showed that you could stabilize the spike protein by mutating the regions in this HR1 region, where you add in 2 proteins, and now Jason has a hexapro, so it's many more proteins in this region that lead to a pre-fusion locked position, locked spike. So it doesn't convert to its post-fusion state. It stays in the form that's really on the virus, on the virion surface, which is important for antiviral development. What's also in the spike protein of SARS-2 is a furin cleavage site, and what Novavax found through a bunch of in vivo studies in mice was that the killing of the furin cleavage site in the sequence makes a much more stable particle and a very strong immunogen in this model. And so we started working with them on their vaccine work, and this is just showing you some of the early work we did early in 2020, where mice were vaccinated. These are biopsy mice. They were vaccinated with and without the adjuvant, prime boost strategy, day 0, 14, and 28 for bleeds, and then challenges. At the time when we did the studies, we only had the adenovirus model, which is really pioneered by Stanford Frieman for MERS many years ago, and used again now for SARS coronavirus 2, where a human adenovirus expressing the human ACE2 gene is used as an intranasal inoculation. It infects all the cells it can in the lung, and now allows for permissivity of, in this case, it was the original Wuhan 1 isolate or Washington 1 isolate for SARS coronavirus 2 that we used in the experiments. Okay. And what was found, was that these, we compared single doses and 2 doses of vaccine in these models. This is ELISA data showing on the right-hand side at 1 dose, as you did a dose curve of antigen with the same amount of matrix M1, the adjuvant, you could certainly see a dose-dependent increase in anti-spike antibody by ELISA. But when you added 2 doses, now you got this really heightened response, upwards of 1 and 10 micrograms per dose. You started to see a really strong response at day 21 and 28 post-vaccination. So we did live virus neutralizing antibody titers on these off of that serum, and we could show that there's really potent neutralizing antibody effects seen in this model, or in this mouse experiment. 1 dose showed low-level effects, similar to what we saw in ELISA's, but the 2 doses really showed a potent and high levels of neutralizing antibodies in mice. We then challenged the mice, as I said, with SARS coronavirus 2, the Washington 1 strain. And when we looked in the lungs after infection, we could see that there was a dose-dependent decrease in the amount of virus in the lungs, really almost sterilizing immunity, where at the highest dose of vaccine, we could have no detectable virus that we saw in the lungs. 1 dose, you certainly could see an effect, but 2 doses, you really completely quenched the amount of virus that was detectable, down to 0.01 micrograms of spike, other than that 1 little bugger of a mouse right there. Importantly, when we looked in the lungs, we saw that in the placebo-infected mice using this model, there's strong inflammation. When you looked in the vaccinated mice, there was certainly still some inflammation around the bronchi, but there was very little inflammation outside of the bronchi. This is 7 days post-infection compared to wild-type mice. So you could certainly see a reduction in inflammation, which has certainly been important for driving this, eliminating the effect of infection even when you're vaccinated. There was a whole host of immune correlates that were done on these mice as well. And so there was looking at multifunctional T cells by ELISpot and flow, really finding there's an induction of interferon gamma positive and TNF alpha positive CD4 T cells and interferon gamma secreting cells after vaccination in mice. There's certainly as well an induction of germinal center B cells being produced in mice, which isn't often seen in protein subunit vaccines. And so we think the adjuvant is really driving this effect in the germinal center development, as well as on TFH CD4 positive T cells, showing a strong enhancement as well after vaccination. There were other animal studies done. This is showing some of the data from a baboon study where baboons were immunized at day 0 and day 21 with bleeds across the time course. Excuse me. Again, showing a dose-dependent response at either 1, 5, or 25 micrograms of the vaccine with adjuvant, or 25 micrograms alone. And so there's a dose-dependent response and time-dependent response in the amount of anti-spike antibody produced by IgG, and then by live neutralizing antibody titers again showed this development, strong development of neutralizing antibodies, even with the low 1 microgram dose of this vaccine. Again, the same similar antibody level or immune correlates were performed in the baboon studies, showing that you, excuse me, showing that you saw a high level of development of interferon gamma secreting cells by ELISpot, as well as interferon gamma, IL-2, and TNF alpha positive T cells, CD4 positive T cells across the vaccinations. So in an additional model that's been developed over the pandemic, the cynos from cynomacaques were also vaccinated, which was published late last year in Vaccine. Again, showing that in the same time course, day 0, 21, and 28, or day 0 and 21 vaccinations, so 3 weeks apart, you could see a high level of anti-spike antibody by ELISA, as well as by live neutralizing antibody. And importantly here, we had a whole host of convalescent sera from patients that were infected with SARS-CoV-2, and the amount of antibody produced in the both the, I didn't point it out, but in the rhesus model, as the baboon model, and now macaques, after vaccination is much higher than what we see in convalescent sera. In these studies, there was challenge that was done in the macaque model, and when you look in the BAL fluid for subgenomic RNA across the vaccination groups, you can see a dramatic decrease in protection of infection in this model, as well as in nasal swabs. And then this model, this was also done in rhesus, as I said before, and studied for the immune response that was seen across across all of the antibody profiling that had been performed. And in work in collaboration with Galit Alter, there was what she calls a system serology panel that was performed, looking at really the serum that's produced during vaccination and what it is about the serum that's unique and what's driving protection in this model. And this was recently published in Cell Reports with Galit and her group. And what she was able to do was perform a whole host of Luminex assays, really trying to understand this large panel of types of antibodies that are produced during vaccination and challenge. And she's been able to find some really kind of intriguing things and weighing what are the key factors that are important for protection. And so in her system serology assays, she will show that if you look in this whole platform for the amount of neutralizing antibody titer that's shown across these metrics, you can see they certainly are upwards on the top of all of these panels that were assayed that were showing really high metrics for protection and leading to a mechanism of clearance. What was also found across all of these was this role of the Fc gamma R2 receptor, which is kind of intriguing, trying to understand exactly what it is about the Novavax vaccine that's important. And in future studies that we're working on, we're working with her now, and she's doing across really all of the vaccines, Novavax, AstraZeneca, Moderna, and Pfizer vaccines, trying to understand the role of Fc gamma receptor and Fc receptor engagement across all these vaccines and how that plays a role in clearance. And the idea behind all these studies is really trying to understand how we can engage this FC receptor and induce these antibodies better by use of adjuvants and various other vaccines to drive this this subtype of antibodies to really lead to better protection. And so I wanted to make sure I leave time for questions, but what hopefully you've seen is that we've been able to, one, that the rise of variants has certainly led to issues in reduced protection across seroconverted people. That's why we're doing these antibody studies now and vaccine studies to understand how the variants are causing disease. That Brequinar in vitro and in vivo can synergize with EID 20-1, and that really understanding the mechanism of this synergy across this combination and then future combinations with nucleosides and protease inhibitors and other aspects of other therapeutics that we're working on in the lab aids in the development of therapeutics, but also tells us a lot about what the virus uses and needs for replication. Hopefully from vaccine work, you saw that the spike subunit vaccines certainly can protect against SARS-2 infection, and this is proven out in the phase 3 trials that Novavax has recently published. But again, it's important to understand the mechanism behind this protection, and we can use animal models to really understand what it is that is driving this protection. And certainly the level of variance, as we've seen for the Delta variant, is leading to a big issue with spread of the virus among both unvaccinated but also in vaccinated people. And by understanding another work I didn't show you, we're looking at things that are outside of spike. So not just the mutations that are in spike in the Delta lineages, but things that are outside of spike across all the variants that are leading to disease. And we've been able to identify several non-spike alleles that are driving pathogenesis, at least in our mouse model. And we are hopeful to understand exactly what's going on in humans as well by studying that response in humans too. So there's a large number of people that have been involved in all of these studies. Everyone in the Frieman lab has really been a big component of driving all this work in the past 2 years. All of the initial drug work was done in vivo, was done by Robert Johnson. And oh, I left Jeremy off here, sorry. And Jeremy Ardenay, who have been driving a lot of the drug work in the lab. Rob helped and did a lot of the vaccine work with Novavax. But really the whole lab really helped out with all of these studies, including Jimmy Logue, who does most of the neutralizing antibody studies that I showed you. That was funded by the Gates Foundation for the work with Sarah Cherry for the Brequinar study, and Novavax funded all of the Novavax work that I showed you. We've been able to get a lot of reagents from friends and colleagues, and thankful to all of our funders that have driven all this work. So I'll stop there and take any questions. Thanks.
Thank you so much. Thank you so much, Dr. Frieman. That was really interesting. Thank you. So as Shinhei has mentioned at the beginning, and you have also a message from Kevin, you can post your questions on the Q&A. So do we have some questions?
So far we have not.
Yeah, so
it was so clear, there's no questions, it's fine.
No, but they will come. You see, that's really the problem with these Zoom and webinars. I mean, here it goes.
So it is coming.
Yeah,
Can you see the question?
I see the question. So the first one up is how does this work compared with baricitinib? So we've actually, I didn't show you all the drugs that we did that didn't work in mice. We've done, I don't know, about 3 dozen or so drugs in our mouse model. Baricitinib we find to be not effective in vivo at all. We've done PK on baricitinib and all of the drugs we're testing in the lab. Baricitinib doesn't show an effect. It actually, if I remember right, it does worse in the mice than what we've seen, at least in the mouse models we've been working on.
Oh, so then that would mean that this really points, but this is not new, of course, but it would really point to the say divergence between these mouse models and human, because there have been some recent results which shows the potential of baricitinib, if I'm correct.
Yeah, I mean, I think a lot of it is timing as well of treatment, and so you know, there certainly can be different PK in humans and mice, and when you give the drug for treatment. So you know, there certainly may be positive aspects. There's only one study showing a positive trial effect for baricitinib, if I remember right. But yeah, I mean, the mouse model can't tell you everything, but hopefully we're able to understand it.
you have another question. So we have actually two questions. One was posted on the chat. I don't know, Shinhei. I mean, some questions are arriving on the chat, the others on the Q&A. I'm a little bit lost.
It's supposed to be Q&A. Probably.
Yeah, but there is a question. Can you see the question on the chat?
I can see the questions. Yeah, I'll start with Alan first. So Alan said, Dr. Bullet, insofar as no antibody enhanced replication or enhanced disease has been observed for SARS-2 in humans. So if you look back, so the question is there antibody dependent enhancement in vaccines that we've seen at all for vaccination? And there has not been any enhancement that anyone has shown or any antibody dependent increase in pathogenesis. In the old SARS-1 and MERS models, you really had to drive the model to the animal models to get enhancement at all. What's really seen is you get eosinophilia that's produced because of enhanced inflammation, because you get non-neutralizing antibodies. When you vaccinate against spike, you don't see that at all. And the only models where you saw that really were where you expressed either N alone or the entire virus as a doubly inactivated virus or as a vaccine. And I think the key point there is that you, for normal ADE effects, like for dengue, you have to get infection in macrophages, and SARS doesn't replicate in macrophages at all. That anyone was able to show significant replication. You certainly can get entry into macrophages and uptake, but it doesn't replicate, and you really need that to drive pathogenesis, at least what I've seen. Franco has a question. Usually mice are not a good model unless express ACE2. I would certainly disagree with that. I think that the mice that the mouse adapted virus, MA10, or the one from Ralph or from Stan Perlman, certainly replicate well in mice. They only stay in the lungs, unlike what's seen in the ACE2 overexpressing mice. And the variants replicate incredibly well in the mice. And I think importantly for the mouse model development, you don't want spread in every organ, which is not seen in humans, at least at appreciable levels. You really want to understand what's going on in the lungs. And I think that in my view, I don't know what the angle is there, but in our view, the mouse model is certainly incredibly useful for understanding pathogenesis and disease. Maria has a question. Do you think all the new mouse adapted variants are lab escapees? I don't see how that's possible. So I'm not sure but that's not at all what we're seeing from any of the variant work that we know of. I'm wondering if you could share your thoughts about Brequinar in the clinic. So the Priya's question about using Brequinar and DODH inhibitors in the clinic for leading to some levels of toxicity. And I think this is an important question. It's certainly something that has to be considered when we use this in, if it's being used as a SARS-2 therapeutic. Usually these are done in the DODH inhibitors are much more longer term treatments. The idea here is for the use of SARS-CoV-2 and any of the therapeutics, including molnupiravir and Brequinar and other nucleoside analog, is that they are short-term treatments. And so the idea is that because of their short-term treatment, you'll have much less off-target and toxicities that are there. And so that's at least the idea, but certainly has to be checked in all the clinical trials to know that beforehand.
Thank you. And so we do have, if I remember correctly, two questions on the chat, Shinhei.
I mean, Dr. Frieman already answered them.
Yeah, that was the same one.
There's another one. No, there is another one from Maria Salva. Do you think all the new mouse adapted variants are lab escapes?
Yeah, I said before, I don't.
Sorry, maybe you answered already.
No, it's okay. I don't know. I don't have any reason to think they're lab escapees or any reason to think that is possible. The lineages are very clear. They all are derived from normal evolution of this virus that we've seen if you look at the phylogenies.
Okay. Thank you so much, Dr. Frieman. Your work really points also to the and it comes as no surprise to the need to have combinations of antivirals over the next years when we will address this type of viral infections. And also to your point regarding the, I mean, there is a kind, not a controversy, but I would say a discussion regarding the molnupiravir as a nucleoside analog, as to the possibility of, say, mutagenesis in humans. And there are some voices as to maybe the authorization in the future might be to some group of patients. What is your take on this?
So, I mean, I'm a cell biologist. I think that the human studies certainly have to be performed and done safely so we can mitigate any of those issues for toxicity across any of the drugs. And because it is a known mutagen, obviously these questions are there to study, and we can study that in vivo as much as we want, in cell culture as much as we want, and in mouse models. But certainly that's certainly a question for all of the human studies as they go on. Molnupiravir has been in trials. I'm sure that they are doing toxicity studies and mutagenesis studies in the human samples now to understand what's going on, and if this mutational event is a problem for treatment with this kind of drug. I mean, I think that it's certainly able to think that, this is common for a lot of drugs, and by studying their mutational landscape is important. I think the hope is that you use this in a short term, either prophylactic or early therapeutic phase, so that you can have strong antiviral effects and mitigate any of the long-term effects you have from mutagenesis because of extended treatment.
Thank you. I believe that we do not have further questions, Shinhei.
No.
No. So thank you so much, Dr. Frieman. That was really a great talk. And we wish a great day to everybody. Bye-bye.
Thank you very much.
Thank you. Bye.